Good afternoon. My name is Filippo Vairo, and I'm an associate consultant at the Department of Clinical Genomics and the Center for Individualized Medicine. I would like to welcome you to the Rare Disease Speaker Series, which is a joint venture between the Center for Individualized Medicine at Mayo Clinic and the Mellowes Center at the Medical College of Wisconsin. This lecture series is an opportunity to highlight the work being done around rare diseases. And it's my pleasure to introduce today's presenter, Dr. Natalia Gomez-Ospina. Dr. Gomez-Ospina is an assistant professor at Stanford University and a board-certified medical geneticist. As a physician scientist, she brings her passion and expertise to the clinic and the laboratory, focusing on diagnosing and treating genetic diseases, particularly lysosomal diseases. At Lucille Packard Children's Hospital, she leads the enzyme replacement therapy and Stanford's program for inherited metabolic disorders, where she works to advance gene and cell-based therapies for genetic diseases. Today's presentation is titled Improving Hematopoietic Stem Cell Transplantation for Lysosomal Diseases, Combining Genome-Edited Cells with Novel Conditioning Approaches. Many lysosomal diseases, especially those predominantly impacting the nervous system, lack effective treatments. While hematopoietic stem cell transplantation holds promise for treating several of these disorders, its broad use is limited by the requirement for donor-derived cells and the difficulty in transporting these cells across the blood-brain barrier. To address these challenges, Dr. Gomez-Ospina's lab has developed novel techniques to modify patient cells and efficiently recruit them to the brain, enhancing the potential of these transplants for treating lysosomal storage disorders and other metabolic conditions. Throughout this talk, please use the Q&A in Zoom to submit your questions. We have planned some time for Q&A at the end of the session. And now, please join me in welcoming Dr. Natalia Gomez-Ospina. Thank you.
Thank you for the invitation. As Philippe mentioned, I am a physician scientist, and what I'm interested in is trying to leverage the immense potential that I believe bone marrow or hematopoietic stem cell transplants have in treating non-hematological diseases, but particularly lysosomal storage disorders. I have a few disclosures, but so I received support for profit and nonprofit organizations, and I have one commercial relationship that's not relevant to this presentation. But I guess an important disclosure for this group is I know that some of you are researchers and clinicians. Most of the data that I'm going to talk about is very early therapy development. So we're talking about cells and mice and a clinical development program that is going to reach patients soon, we hope, but no clinical testing yet. But we'll review some of the things that have been tested clinically. So I'm going to try to give you a whole overview of how transplantation works for these types of diseases. So for these non-hematological indications, right? It's very unusual that we're transplanting blood stem cells to treat disorders that are really not blood disorders. And I'm going to spend a lot of time talking to you about the role of pre-transplant conditioning in these indications, because it is 80% of the equation of why we can get these bone marrow transplants to affect the brain, the liver, and other organs. And it's also part of the part of the procedure that is so feared by clinicians. And so I'm going to tell you a little bit about it, what it is, how it works, why it is important, and what the work that we're doing to make it safer and more effective. And then I'll spend the next half of the talk talking about autologous transplantation using genome editing to treat these disorders. And we're going to focus on the two programs that are most advanced, MPS I, or mucopolysaccharidosis type 1, and particularly for Hurler syndrome and for Gaucher disease type 1. So bone marrow or hematopoietic stem cell transplantation has been used for decades to treat several genetic neurodegenerative diseases. It is this HSCT, hematopoietic stem cell transplantation, is the standard of care for multiple inherited metabolic disorders, including several lysosomal storage disorders, peroxisomal disorder. Some of these have primarily neurological manifestations, some have systemic manifestations that include the CNS, but also other organ systems. HSCT is also a very promising investigational therapy for several neuronopathic and non-neuropathic LSDs, and multiple studies suggest that it could be beneficial in many other diseases. So this table is very busy, and the point of the table is a little bit to shock and awe you about all of these possibilities of diseases where there's either we use it or there's promise that it could be used. So in some of these diseases, we're conditioning and transplanting these children. So what we're trying to do here is to make transplantation safer and more effective and to reverse the risk-benefit ratio so that it becomes a feasible approach for some of those indications down here at the bottom, where there's proof of concept and even a theoretical benefit. So how transplantation addresses disease pathology in non-hematopoietic tissues is really not well understood. And here we're going to focus on the brain. How it halts neurodegeneration is not understood, but the effect we know is mediated in part by these cells that migrate from the bone marrow into the brain to become tissue-resident myeloid cells, or where they can replace microglia cells, which are the immune cells in the brain. So once they're in the brain, they may replace a microglia cell that may be defective or may decrease inflammation that way. But generally, the way that we understand this is in lysosomal storage diseases, these cells that are coming from the bone marrow are able to produce missing enzymes that are then secreted or can be uptaken by other cells that need it in a process known as cross-correction. This migration or repopulation, people refer to it in different ways, is determined in large part by the conditioning regimen, which is required prior to transplantation. And again, there's a lot of things that we don't know about this, the cells that are involved, the role of blood-brain barrier, and the role of other cells. So, and as we've learned, as we've done this process more and more efficiently, we're calling this a process of microglia macrophage replacement, and I'll show you why. So there are two important limitations to the current use of transplants for these indications. The first one is the use of allogeneic cells or donor-derived cells. These can predispose a patient to immunological complications since you are inheriting somebody else's immune system, can delay treatment while you search for a donor. The other one is the transplant conditioning, which is associated with significant morbidity and even mortality from the direct toxic effects of chemotherapy. That is what conditioning is, as well as potential infections and profound immunosuppression. So, and right now, the way that we do it, the conditioning really has poor CNS activity. So it doesn't allow a lot of the cells to go to the brain and is very slow in recruiting cells to the brain, and it has this generalized toxicities. So what we envision for the future is we can use autologous cells, the patient's own cells, to decrease immunological complications. And that means developing very efficient cell modification strategies to do this, because if the patient has a genetic disease, you have to correct it one way or another. And to try to develop near conditioning regimens that are better at CNS conditioning, that can work much faster and much safer. And I'm going to address the problem in conditioning first. So, for most transplantation, you have to condition, meaning you can't just infuse stem cells into a patient and they will just engraft and stay in the body forever. We understand the role of conditioning from the point of the bone marrow, and that in the bone marrow, it's necessary to clear the stem cell niches for these new stem cells to come and take up residence. It's as if the stem cells lived in these very special places that need to be clear for the new cells to take home and to come and engraft. In the allogeneic setting, like we'd use that, which is the cells come from a different donor that is non-genetic, is genetically different. The condition also provides immunosuppression to ensure to ensure engraftment and to prevent rejection, because if there are cells there from the patient, they may reject the graft. Now, if you look at this arrow on the right side, conditioning can be deployed at different flavors, so minimally invasive, reduced intensity, even myeloablative. And as you increase the intensity, you increase the toxicities, but you get better engraftment. So chimerism, meaning the percent of cells that come from the bone marrow and the donor, will be much higher the higher the intensity of your conditioning. Now, again, when we condition a patient for a bone marrow transplant, we don't really know what's happening in brain and other tissues. We know that recently studies suggested that when you use busulfan, which is the agent most commonly used in the clinic, at least for these indications, that it induces microglia senescence. So at least in mice, when you look at mice, you give them busulfan, all of a sudden the microglia senesce, they change their shape and they stop dividing completely, which is a property the microglia have in response to certain stimuli. We also know every time we talk about this, people think, well, it's a blood-brain barrier opens up and cells go in. The bone barrier doesn't open up. It stays, at least studies of blood-brain barrier permeability have shown that there isn't a major increase in permeability due to conditioning. And we also know that for it to work in these indications, it has to be myeloablative. So we have to give these patients a high dose of conditioning, and the conditioning has to act on the CNS. So either have to do radiation that involves the head, or a drug that actually crosses the blood-brain barrier. And I'm going to show you some data about that. So as we do it today, transplantation has many limitations, a low engraftment of these bone marrow-derived cells in the CNS. In fact, it's so low that for not till recently is still somewhat disputed by some people whether these cells actually go to the brain or not. It also has very slow kinetics of repopulation, which is a limitation, especially in rapidly progressive diseases, like some of the lysosomal storage disorders. And there are many genotoxic and side effects of myeloablative conditioning. So we and others thought, okay, maybe there's a new drug we could use or a new target to condition. So we thought about this receptor called CSF1R. So, and the reason why we thought about CSF1R is if you look at this panel C, there's this CSF1R knockout mouse. It lacks, CSF1R is a receptor required for microglia and macrophage survival and proliferation. So without that receptor, the mice have no microglia, no macrophages, no osteoclasts, no macrophages in any tissue. They can be rescued with a bone marrow transplant. And when you give the transplant, you see that this is transplant of green cells, you see that the whole CNS is now entirely repopulated by cells that came from the bone marrow. They were originally hematopoietic cells, but they've been able to replace the microglia in the setting where the mice had no microglia. So fortunately for us, we said, okay, can we use these drugs? Fortunately, there are many inhibitors of this receptor that have been used, or actually one of them is FDA approved, this one, the PLX2397, to inhibit tumor-associated macrophages. And so the nice thing about these receptors is they cross the blood-brain barrier, and they're actually pretty safe to use. They're safe in mice and humans. Humans, PLX3397 is approved for tenosynovial giant cell tumor, and I don't think it's being tested for glioblastoma. And patients take it for prolonged periods of time, and if the side effects are minimal, there certainly is not the side effects of chemotherapy. And I'm showing you in this schematic from this paper showing that the effect is reversible. So you take mice and you see the green dots of the microglia cells. They take the drug for 28 days. Then you stop the drug and the microglia slowly come back so that by 2 or 3 weeks, the microglia have come back. So they're reversible. They have a pretty reasonable toxicity profile. So a long story short, we've learned that we couldn't just use these drugs to condition by themselves, which would have been great. But here's what we found. If you combine them with standard condition approaches, so here's a mouse that was conditioned with either busulfan, which is what a patient would get, total body radiation, which is what mice normally get in the lab, and treosulfan is a cousin of busulfan that doesn't cross the blood-brain barrier. You give them a transplant with the green fluorescent cells, and then a few days later, you give them just a few days, 5 or 6 days of this oral drug and see what happens. And we measure by flow cytometry, where we can measure microglia by these two markers, CD45, CD11b, and we see what percentage of them are actually GFP positive, which means they have to come from the bone marrow. So when you look at the bone marrow, you see the bone marrow is all entirely from the donor. It's all entirely green. But when you look at the microglia, busulfan alone gives you this tiny little bar of 6%. Busulfan plus a few days of CSF1R inhibition gives you 90%, so 15-fold increase over the number of cells that were there. TBI gives us 30%, also maximized by giving CSF1R inhibition. But when you use treosulfan, which doesn't cross the blood-brain barrier, you don't see anything. And so which means that, okay, you need some conditioning of the brain using a standard chemo, but when you use this a few days of inhibitor, you can really see the increasing the replacement of microglia. And if you have a trial, and if you follow this long-term for 8 months, this is a persistent long-term effect of having replaced the microglia, where there's still 90% of the cells come from the bone marrow. It's still much higher than busulfan. And this isn't, there's not like a lot more cells go to the brain. T cells, which are labeled by CD3 marker, we don't see them. There are no B cells, which are labeled by the CD19 marker, and not other inflammatory monocyte cells or macrophage cells. So very specific recruitment of a certain type of cell. And if you have trouble imagining this, so here's the sagittal section of a mouse brain, with every cell that came from the bone marrow, it's labeled in green. And they have this nice sort of ramified morphology. So these are all bone marrow-derived microglia cells, which we call microglia-like cells because after many studies, we know that while they have changed their identity from having been from the bone marrow to become microglia, they're not exactly microglia, and that they don't have this sort of embryonically distinctive microglia genes, but they perform many of the microglia functions. So we know these cells can go to the brain. We now have a very safe way to increase the number of cells that go to the brain. And we also know that this process happens so much faster. So if you are a transplanter or you've ever treated these patients, we know that we usually like to treat these patients very early in the disease process, ideally even asymptomatic, because it takes a long time for the transplant to take place. And that's because the migration is really slow when you do it using the standard conditions that we do right now. So this is probably what's happening in a patient's brain. Very slowly, after a couple of months, you start to see a significant number quantifiable of cells in the brain, which can be sped up significantly, 20 times faster by adding the CSF1R inhibitor, which is a very important part of being able to treat, again, a rapidly progressive LSD. So getting cells there faster. And we did a lot of work to show that this is safe. The adding just a few days of inhibitor is safe, doesn't affect bone marrow reconstitution, doesn't affect the cells, doesn't affect the behavior of the mice, doesn't affect the function of the cell. So the whole point of these 13 minutes of talk was to demonstrate to you that cells from the bone marrow do go to the brain, and you can envision these cells as cell therapy factories, live therapy factories that can be used not just for lysosomal storage diseases. So microglia depletion, meaning a combination of busulfan with CSF1R inhibition, can enhance the efficacy of these transplants by enhancing the repopulation of these bone marrow-derived cells, by speeding the kinetics of repopulation. And as I've shown you, these cells are very much similar to microglia cells. So now that you understand that part of the transplant and how we can make them better, let's talk about making autologous transplantation. So the idea of self-transplants is not new. This has been in development for decades. Autologous transplantation of hematopoietic stem and progenitor cells, HSPCs. There are already 2 products that are approved. You can read the names. They're really long. One has just been recently approved for metachromatic leukodystrophy, just last month in the United States, was previously approved in Europe. And another one has been approved for adrenal leukodystrophy. So these patients will receive our current standard conditioning, which is busulfan-based, not the CSF1R inhibitor until we get it in clinic. But they're going to get their own cells, which have been modified, what we in the gene therapy field call non-targeted gene addition with lentiviruses. So these lentiviruses package a copy of the gene with a sequence that drives expression with a promoter. And these lentiviruses will insert themselves randomly or pseudo-randomly into the genome. So they'll be part of the stem cell. As being part of the genome, they will be part of every cell that comes from that stem cell. As they proliferate, as they differentiate, as they make their progeny, it'll be part of the cell, right? And so there's fantastic long-term data on the safety of this for these that have been approved, and also for other ones that are in development. But there are still some concerns about this sort of pseudo-random integration. And in fact, for the XALD therapy, there is a warning about hematological malignancy, and that a certain number of patients were developing life-threatening cases of myelodysplastic syndrome. So this may be vector specific, and I want to highlight that I love the work the lentivirus field has done on this, as we, genome editors stand under the shoulders of our work, but there are still some concerns. And now we have genome editing, which enables us to do very precise modifications, so do a very targeted modification. So despite tremendous progress in this gene addition using viruses, precise genetic modifications can be a better option for achieving very predictable patterns of expression, or even targeting specific alleles, which is what you need to do sometimes. So genome editing, as you know, is a mechanism to rewrite DNA sequences. And typically we think of it as a way to correct a mutation back to the correct sequence. But in fact, it can be used to create new sequences that sometimes can have more therapeutic value. And I'll give you an example of that. So the core of the genome editing, most commonly talked about genome editing-based platforms, are the nuclease-based genome editing, which at the core of them is sort of a nuclease that you were able to guide to a very specific site in the genome to cut. It's how you guide that nuclease to cut that these different technologies differ. So one of the first ones were the zinc finger nucleases, where the mechanism was having a FOP1 nuclease fused to an array of zinc finger repeats, and those zinc finger repeats dictated which sequence this nuclease was going to bind to and where it was going to cut. This required a lot of engineering, and so it was really hard to do. But nevertheless, this was the first genome editing approach that was tested clinically. You can see this announcement in Science for this patient that received a zinc finger nuclease treatment, a patient with mucopolysaccharidosis type 2 and MPS2 attenuated back in 2017. So There were two clinical trials, the Champions trial, which was for MPS II, and then Powers trial was for MPS I, and was an in vivo, so the zinc finger nucleases were injected in the patient intravenously, and they were supposed to go into the liver, and in the liver, they were supposed to modify the albumin gene to make the missing enzyme. The data, the trials showed that the therapy was safe. They didn't have any serious adverse side effects, but it wasn't effective. So it wasn't, the editing was too, wasn't efficient enough to have an impactful biochemical result. Since then, we've had the sort of most spoken about platform, which is the RNA-guided nucleases that are derived from the CRISPR-Cas9 system. This was a mechanism that provided acquired immunity to bacteria. Now, for research and therapeutic purposes, we use a modified or better said engineered version of the system that is basically two components: a guide RNA, and a CRISPR-associated nuclease, which is the Cas protein. So the role of this guide RNA is to guide the nucleus to the target site. And because this RNA-based recognition, targeting different sequences only requires changing the RNA, which is so much easier to do than engineering proteins. So this versatility really drove the widespread and rapid adoption of this technology for basic research and therapeutic purposes. So after the double-strand break, the cells recruit two different repair mechanisms: non-homologous end joining and homology-directed repair. They both have been used therapeutically. Now, if you've heard about the CRISPR therapy for beta hemoglobin, for beta thalassemia or sickle cell disease, relies on therapeutic NHGJ. Where I'm going to talk about is the use of homology-directed repair for these uses. So, okay, if you're going to use genome editing again, you can use an in vivo approach, like it was tested for the in vivo for the zinc finger nucleases, where you package the genome editing components into something, generally use a virus, and inject it directly in patients, or you use an ex vivo approach. And for that, you need to have, again, a cell that has regenerative potential. You can extract it, you can culture in a dish, you can modify it, and you can put it back, and you know certainly that when you inject it back, it should reconstitute that organ system. But despite a lot of work on iPSCs, the hematopoietic stem cells, we have 4 decades of experience isolating them, transplanting them, and modifying them. So this makes a great sense as a target cell. I'm on the field that ex vivo offers certain advantages, is that you can characterize your product, your off-target toxicities, before you put it into the patient. And so we are adopting an ex vivo approach. So the approach is as follows. We isolate the patient's hematopoietic stem and progenitor cells. They have a marker called CD34 on the surface. The cells are modified ex vivo using CRISPR-Cas9. We use a safe harbor approach, and I'll show you what that means. And the idea is that once these cells are modified, we transplant them back in the patient. They make tons of enzyme or whatever we want. They reconstitute the bone marrow. We reconstitute those macrophages. We reconstitute the microglia, given the right conditioning approach. So we use a safe harbor, and the safe harbor is we use CCR5. CCR5 is, we think it's a safe harbor because there's a great percentage of people in the UK Biobank that don't express CCR5. CCR5 is the receptor for the most common form of HIV. And it was discovered that people who didn't express the receptor were resistant to HIV infection. So you don't need CCR5 to develop and function normally. Your immune system doesn't need it either. But the advantage of a safe harbor means is that you can control the gene expression, which sometimes is important. You can control, it's a single solution to say allelic heterogeneity, which means like for certain diseases, there are hundreds of mutations. So you can't have a genome editing approach for individual mutations because you have to design it for each patient. So this allows you to have a universal approach for all of the mutations in one disease. It allows you to design the coding sequences, meaning that you can actually engineer the proteins to make them more stable across the blood-brain barrier. There are all kinds of new applications for lysosomal enzyme engineering, and ultimately is adaptable to other diseases, especially when you're talking about lysosomal enzymes where they have a similar mechanism. So you could just adapt it to the next enzyme and hope that it works very similarly. So exactly how do we do this homology-directed repair with CRISPR-Cas9? So I think I mentioned we use basically 3 components. We have a recombinant Cas9 protein nuclease a synthetic guide. This is a chemically modified guide that we discovered was better than just any RNA. And so these combined, the guide will guide the Cas9 to the CCR5 exon 3, where it will create a double-stranded break to trick the cell into inserting an enzyme expression cassette. We provide the cell with the template for repair in the form of an AAV6 genome. This template for repair has homology on both sides of the cut and has a cassette in the middle, so a promoter and an enzyme that is tailored for each individual disease. And after the cells do homology-directed repair, then that cassette gets integrated into the CCR5 locus. So I'm going to show you the data, the development data for this therapy for MPS. So mucopolysaccharidosis type 1 or MPS 1 is sort of your model lysosomal storage diseases for testing new therapies. I chose to start with this because it's a multisystemic disease, as you can see in the symptom table. There's cognitive impairment, change of facial features, there's joint involvement, visceral involvement. A lot of organs are involved. So it allows to test the efficacy of your therapy in all these different organ systems. Like many enzymatic deficiencies, there is sort of a continuum of activity for patients with severe MPS 1 or Korler syndrome. If you look down here, the pink box, that is sort of the average enzymatic activity of the patient fibroblast. So for you to have severe enzymatic activity, you have to be really, really close to zero for severe to have disease. Patients with attenuated disease, which have a very different course with less cognitive impairment, still pretty significant involvement of their musculoskeletal system, although they do have some cognitive issues, ADHD and other things. They have some residual activity, but it's not 20%, it's not 100%. It's usually just a couple percentage of normal. And that's important to know when you're setting the threshold for where you want to get to. So in order to fix MPS 1, we don't need the patients to make 100%. We don't need the patients to make 50%. The parents made 50%. But, we need to get them way above a couple percent, at least in the brain, in order to very, very significantly change the course of the disease. And the important thing about MPS 1 too is that it's part of, by now, 44 states in the United States are screening for MPS 1, which allows us also to intervene earlier. So the problem is the disease is the deficiency of iduronidase, and what accumulates when that enzyme is missing is a metabolite accumulation of glycosaminoglycans, it's repeating units of disaccharides. And when these accumulate, this is toxic to cells, many cell types, which results in a progressive multisystemic disorder. So using the system that I mentioned, so in the preclinical studies using mice, we targeted the CCR5 human. So these are human hematopoietic stem cells that we're editing using the system where we insert a cassette that just has the iduronidase driven by a promoter that is called PGK. This promoter is active in all cell types in the hematopoietic system. So T cells are going to make it, B cells are going to make it, and it's going to do it at very high levels, higher levels than normal. And in some of the cells, we edited with fluorescent cassette just so that we could track them, separate them from the unedited cells, and really characterize them, but also track them in the mice in vivo. And the clinical cassette would look something like this without the fluorescent. So in our earlier studies back in 2020 or something, 19, we could get, so if you use hematopoietic stem cells for cord blood, you can get on average about 35% of the cells would be edited if they had a large cassette. And with the small cassette, which is the clinical cassette, we called it here, here on the right part of the graph, you could get about 45% of the alleles were modified. So alleles is different than cells, and I'll tell you the difference between those two. But if you take those cells out, which you can see these hematopoietic cells differentiated into macrophages. These are human macrophages that have been edited. They express about 40 to 60-fold more enzyme than an unmodified macrophage or an unmodified microglia, or even an unmodified HSPCs. And a concern when using genome editing is the potential to create off-target edits in a place that you don't want, right? Because you're creating double-stranded breaks. So one way that we address this is to actually use a program to predict where the off-target sites would be. And here, these 67 off sites that were predicted, we deep sequence all of those potential off-target sites. And then at that time, we were working with IDT on developing a higher fidelity Cas9, and we did the expansion of those. So wild-type Cas9, red dots, when we used the wild-type Cas9, we found that of the 63 sites, they were the on-target activity, which is your first dot here, is, or your first, and the first line in the table is 90-something%. So that's where I meant to cut. I cut 95% of the alleles. That was what I meant to do. And then the 1, 2, 3, 4, 5 potential off-target sites that came up that were revealed when we used the wild-type Cas9, all of them you can see are less than 0.5% and reaching the level of detection of the sequencing. And most importantly, when you switched our assays to the high fidelity Cas9, those targets went away, and we don't see any off-target activity within any sort of exonic sequences. In order to editing human cells is great, but in order to test the efficacy, you have to have different animal models. So we made a new mouse model that actually didn't have an immune system, that had MPS1, so we could engraft the human cells. So this is a model of MPS1 that lacks an immune system, is available in Jackson laboratories. The same that doesn't make hyaluronidase. It has accumulation of GAGs, and it has skeletal dysplasia as well, with some of the features that you'd expect from PS1. And then now you have the cells, the human cells, the edited human cells, and the human engraftable mouse to do these studies. So you can see that here, and then in these experiments, we're comparing busulfan versus troll body radiation. And with myeloablative busulfan, we get much better data, which is the dark red. You see higher levels of hyaluronidase reconstitution in the serum, in the liver, and the spleen. In these models, we get 1,000-fold more enzyme in these tissues because of the property of the xenotransplant model, where the human cells love to go to the spleen. But you can see that also the urinary GAGs are much better, mostly normalized when you use busulfan, and also in the liver and the spleen. Now, in the brain, what happens? So this is an interesting experiment. We do get enzyme reconstitution in the brain that, again, is better when you use myeloablative busulfan. You get some statistically significant improvements in the levels of GAGs, but you don't get a lot. And that's partly because human cells, I showed you those beautiful pictures of mouse cells being recruited into the mouse brain. Human cells are now well recruited into the mouse brain because they don't react to the same chemokines, particularly the MCSF. Mouse MCSF doesn't respond to human MCSF. But you can still see these human cells, which are labeled here with these green dots, surrounding the brain and occasionally within the parenchyma. But for this disease, that's enough for us to say, here there's some improvements. And so with this data on feasibility, efficacy, and safety, we were able to apply for an award from California Institute of Regenerative Medicine, which is a tax-funded agency to fund stem cell research. And they've given us the money to conduct the IND-enabled studies to proceed with this therapy. Those studies are basically, can we manufacture it at a clinical scale in a GMP-compatible process? Can we do additional studies on safety, including like in vivo tumorigenicity studies? And so we actually are at the point we have a clinical product. So we work in a GMP facility. We've optimized, once you go from the lab to the GMP facility, it's a totally different process, right? You have to figure out, question very, very carefully, do I need all of these reagents, all of these things that I use preclinically, all these cytokines, do we need them? How are we going to grow them in flasks? And how are we going to we use an electroporation to electroporate the guide and the nuclease. And so we had to optimize all of these factors to get to a clinical product. So we call this therapy GT-CCR5 as the first of the CCR5 platform, MPS1. We are able to get very good levels of CD34 enrichment, which our criteria was 80%. Most of the cells are live and very healthy. Our on-target efficiency, we said in order to get clinical improvement, we need to get our product consistently has to have more than 20% alleles edited. And we titrate the doses, the lowest dose of vector genomes that we needed in order to get decent therapy. So an interesting thing I've been telling you about modification of alleles versus modification of cells. So as you can see, you can have, usually the number of the edited alleles is usually lower than the number of cells, because most of the time we have cells that are modified in just one allele. And so when you actually measure the number of cells, the number of edited cells is actually 60 or 42% when we use 5,000 vector genomes per cell of the AAV. So we can modify 60 to 40% of the cells that we get from the patient. And the activity of these cells, we have measured in the preclinical studies, is around 44% of over normal cells and edited cells. We have done additional work in order to increase the editing efficiency of these guides. So we found a guide, so a standard versus an alternative guide that gives us an improvement of about 1.5% in the editing, which makes a big sense when you're making a lot of difference when you're making a therapy. So you go from 40 to 60%. And also we are working with IDT or working on these boosters, which can increase the editing efficiency by 1.4%. So combine those two modifications, we can double our editing efficiency so that we go now from editing 50% of the cells to editing 74% of the cells. So at this point, we have our product, we can officially manufacture a patient scale that meets all product release specifications, and we're expecting to file an IND at the end of this year with a phase 1 clinical trial expected in 2025. So the last few minutes showing you how, because I showed you this is a flexible platform for other diseases. And the nice thing about it's a little bit boring from a preclinical development side, but once we've invested all this money, all these millions of dollars really in understanding the toxicity of the genome editing regions that we developed, that guide with that virus, we can apply that to our next program, because we already know that guide is safe. We don't have to reinvent the wheel. We already know if, we understand it's off target. Anything to speak of. So we're using the next target is Gaucher disease type 1. So Gaucher disease is one of my favorite diseases, probably because I don't quite understand it entirely. It's not just one disease, it's a spectrum of diseases from type 1, which sometimes even patients with 2 mutations can be asymptomatic, to type 2, which is a very severe neuronopathic form, to even hydrops fatalis. And there's increased interest in Gaucher disease, particularly in mutation, or because mutations in this gene, carriers of mutations in the genes have a high risk of developing Parkinson's disease and other synucleinopathies. But for Gaucher type 1, or the non-neuropathic form, patients have hepatomegaly, they have cytopenias from bone marrow involvement, they have bone diseases, osteopenia, lytic sclerotic lesions, osteoporosis, they may have pulmonary involvement, they may have certain malignancies. And this is the most common form in the Western world, not necessarily in other countries. So again, the pathophysiology here is an enzyme, glucosylceramidase, which will abbreviate as GCase. In this disease, this accumulation of glucosylceramide and glucosylsphingosine. Gaucher is very unique in that most of the symptoms are really the result of infiltrative phenomena in the mononuclear phagocyte system, which is meaning that most of the problems, it's the macrophages. The macrophages are the problem. There are already several therapies for Gaucher. This is one of the first rare diseases where there was an effective therapy. So there is enzyme replacement therapy. There are three different forms of enzyme replacement therapy. There's substrate reduction therapy, which also works quite well. And there are other therapies in development, gene therapy, viruses in vivo, AAV-based gene therapy, but also there was for a while ex vivo antiviral therapy developed. There are other chaperones that are being developed. Now, in the past, allogeneic BMT or HSCT was actually used probably in the 80s. It was curative. It could correct the hematologic and visceral and bone problems in the disease. And it could act in the CNS, so it could be used for GDT, Gaucher type 3. But the morbidity was so high that the benefits didn't outweigh the risks. And SRT and ERT has become the standard of care, has really replaced it. But because it is primarily a macrophage problem, you can envision that a safe bone marrow transplant of autologous cells could be effectively curative. So for this disease, instead of what I showed you before for MPS I or iduronidase, the cells had what we call a constitutive promoter, PGK, where all the cells express it. For GCAs, we had a hard time using the same approach because the HSCs didn't love overexpressing GCAs. So it doesn't matter. We could use the same system, the same guide. We just changed the promoter. So we engineer a small promoter that would become effective only in the monocyte macrophages, which is the cell that most needs it. And you can see in the cells that if you compare constitutive promoter with the CD68 promoter, the cells, the HSPCs that have been modified, don't express GFP or the enzyme. Once you differentiate it to macrophages, the enzyme expression turns on. At the end, these macrophages express about twofold more enzyme than a normal macrophage. And you can see this is also a human human HSPC that was differentiated into macrophages that had been edited with this promoter. And we study the properties of the macrophages, and they're normal. Here you can see them endocytosing E. coli. A very important question when you're studying hematopoietic stem cell therapies is to make sure that there's long-term engraftment. And we show these with the serial transplantation studies, where we put them in the mice for 4 months, then we take those cells and we put them in another mouse for another 4 months. And you can see that these cells maintain the long-term engraftment differentiation studies. They're present in the primary transplants, then we do secondary transplants, and they can still engraft. In order to test the efficacy of cells, we have a new mouse model of the disease where we actually especially delete the enzyme in the hematopoietic compartment using an MX1 Cree and a poly-IC induction. These mice don't have enzyme. They develop anemia much faster than any other model before. By 3 months, they're already anemic. By 3 months, they already have splenomegaly. By 3 months, they already have hepatomegaly. And by 6 months, they already have osteopenia. This is a new model that basically had a different genetic background than the ones that are used normally. And so we're able to get much more severe phenotype, a very typical phenotype of GD1. So, and then we can edit the mouse cells with the human cassette and show that about 10% of the alleles after 4 months later are still engrafted. We normalize the anemia, normalize hemoglobin counts. These cells express higher than normal GCase activity, about twofold more than healthy or normal wild-type cells. The splenomegaly is resolved, the hepatomegaly is resolved, and we get the Gaucher cells are pretty much gone after 4 months. So I've shown you with these examples that I've shown you that the hematopoietic system can deliver these therapeutic proteins, particularly lysosomal enzymes, to other organs by replacing this tissue-resident macrophages, and particularly in the brain, by replacing these microglial cells. This safe harbor approach is a flexible platform for the expression of lysosomal enzymes. Or if you're more imaginative, you can imagine, if there's other protein that I think is secretable, it could be expressed by these types of cell types that could be beneficial in the type of organ system that I'm working on, lung, liver. This is also a potential approach. And I hope that I've also shown you that this microglial depletion also enhances the efficacy and safety of HSCT for these neurological indications. And with that, I would like to thank the courageous folks in my lab who joined an assistant professor on all these very difficult projects. I would like to thank all of my funders, especially California Stem Cell Agency, for very significantly funding work on rare diseases and especially stem cell research. And with that, I will take any questions.
Dr. Gomez-Ospina, thank you very much. It was brilliant. This safe harbor approach is super smart, and we are very excited about this. That's great. I'm going to open up for the panelists if they have any comments or questions, while we have some time for questions from the audience as well. So if you have any questions, please type in the Q&A button here in Zoom, please.
A very, very simple comment is that it was just actually a tour de force. I think that it's very important for us to listen to your experience. Obviously, that most of our center works with a different type of rare and undiagnosed diseases, right? And we develop some focus, but we also have other patients going on. And so it's good to know the technology that you have advanced. I think that there was a significant proof of concept. I really loved it. I can't tell you. So I really thank you. I might have a questions which is actually more from the cell biology of the cells. I have done a lot of freeze fracture, cryosubstitutions, and things like that, of lysosomes in my life. And so I have also seen packing of lysosomes and how they pack enzymes and so on. So is there any concern with the fact that there is a very high expression of, not that, I mean, you didn't have anything there that I could see as to worry, but any concern or anything for those who are expressing large amount in the lysosome that could actually alter the sorting of other lysosomal enzymes?
That's a great question. This is one of the experiments that I've wanted to look at just to see. I imagine that maybe not all the enzyme that it's made in these cells is processed appropriately. I mean, we know they cross-correct. Some fraction of it is being secreted, some fraction of it is being active. To me, like the most important aspect of the safety is engraftment is always like my most important readout. So do the cells engraft? Do they differentiate? Unfortunately, we have some idea of how that works in the mice. So we know that they engraft long term. We know that they can make T cells, B cells, and doesn't affect that. Now, that is true for iduronidase. And we know that from there's a lentiviral trial that is using overexpressing iduronidase as well, and they use the same promoter, it's PGK. So at least that's gone into patients, and I don't think there's problems with toxicity.
Okay.
But I don't think that's generalizable to other enzymes. We had a hard time with GCAs. We're trying to work with CRAB-A, and GAL-C is even more complicated. You can't just express it in certain types of cells.
Yeah.
Yeah.
That's a great answer. I think it makes sense. The second question maybe, and I stop there, is that, I mean, this enzyme, obviously, I also, in my life, I have done some few things. So I work on glycosaminoglycans on the surface of the globule of the fat of the milk globule. It has glycosaminoglycan, right? And this enzyme can eat those glycosaminoglycans. And those play a significant role in tyrosine kinase receptor signaling and so on. Any comment that you might do? Obviously, you are trying to cure a disease that whatever, but in the sense of just the intellectual part of the study, I guess.
Yeah, we've always wondered, a lot of the process development, and this is when agreement with the FDA is, we can't process development. I mean, we try things in the lab to get editing right and to get the scale up. We can't do that with patient cells really, because we don't actually have that many earlier patients that we can just get cells to play with, right? So with the FDA, we agree, like you will use healthy patient cells to do these experiments. And so, we pay lots of money to get these cells but we've actually never worked with MPS1. And I'm always wondering, like, the GAG component, the the cells will be different. AAVs bind to GAGs. Like, will there be something different? So I think we're trying to do some small experiments to say, let's make some. The problem make some MPS patient cells by first knocking that out, like do it ourselves, knocking it out. The cells don't like the double editing though, but I think that so there is a very interesting biology now that you ask, which is the conditioning part. It's very disease dependent. So that beautiful data on wild type mice, all the degrees. When you try MPS1 brains, it doesn't work the same. It doesn't work as well because the microglia in those, or whatever, the brain in those animals or in those diseases, it's signaling totally different. So when we measure the cytokines that are released, it's much lower. So of course, GAGs are super important for signaling in general.
Well, I really thank you again. There is obviously those intellectual things will be sorted out as we go on, but this is a tour de force, and congratulations and thank you for coming, and from my part to Mellowes.
Well, much agreed. And I have a little bit of follow-up that you just partly answered already, which is that in some of the disease systems, the replacement is not as high or as fast. And what you outlined completely makes sense, that when you want to be able to act fast. And so to get 90% replacement is just incredible. But I'm a little bit curious, especially for the diseases where you're only targeting, like your therapeutic threshold is a few percent of replacing the wild type function. And because you mentioned, and you glossed over a little bit, that they're glial-like and not fully glial. So when you come into patients, is your goal to kind of more titrate things in? And how are you wanting to approach that? Like maybe we don't want to get 90% because we've lost some of the normal glial function, which long term might be better. Clearly, you got to treat the disease first. But are you aiming to have a little more moderate replacement?
That's a great question. So, I was very excited about this because, again, the drug is safe, it's already FDA approved. And it's like, okay, so how do we move this next? And some of the questions you're saying, like, is it really a good idea to replace night? But now, number one, we do not really know. And how do you even study this? Like, how do you measure microglia replacement in someone who's alive? So I guess on one side, I say, okay, we've been doing it badly for decades. Okay, so we've been doing these transplants on these patients for a while, and we've been getting very few cells, which are still microglia-like cells. Maybe, so they have some function. We also have learned over time as people have become much more comfortable with this. I mean, 5 years ago, we started talking about people be like, oh, microglia are like we derived, that doesn't happen. But now, like, there are people that we're looking, this process happens normally, much lower rate than after a transplant, where bone marrow derived cells go to replace microglia. So we're finding them in these older brains. So I don't know what else it would take to convince someone that these cells are basically functional. We've done single cell and we've characterized them up to the wazoo, and I can tell you they have signatures that are this way. They're they're anti-inflammatory primarily. And there's tons of data where these inhibitors are being now studied about, like, the depletion of microglia has this rejuvenating effect in Alzheimer's brains or Parkinson's brains. So people think these new microglia that emerge could be healthier. So I don't know if I answered your question. I really don't know. And there are these disease-specific differences where it's like, okay, even if I had a perfect experiment of 5 days of inhibition that were perfectly in this mouse, in this type of brain, I don't know what's going to happen really in the MPS I patients. What we see in the mice is maybe in the MPS I setting, we only get like 30-40%, which is still much better than 5%, which is what we're treating them with, right? Maybe that's going to be enough. So I don't know. If you have any ideas, let me know. But I feel like we should do something to increase the efficacy, because already the conditioning we're doing is just terrible for these types of indications. And there's a very powerful tool to improve it, but I don't know. I think maybe we need larger animal experiments.
No, I think that's a great answer. And it's to focus on the primary issue first. And then if there's a secondary, we'll learn about that. But now we can at least address the primary in a more effective way. Thank you.
Thank you.
Dr. Gomez-Ospina, we do have some questions in the chat, but and because we have a very broad audience here, could you share with us your experience of applying the same approach with maybe non-classical metabolic conditions, when it's not like a single enzyme that you are targeting? Is there some ideas for other diseases?
We have some interesting projects. I mean, I'm an assistant professor, I have a small lab. It's not like I've tried every disease. So, we're trying the obvious, Krabbe, some progranulin. There is one interesting indication which brings a very interesting topic into discussion, which is Friedreich's ataxia. We're trying transplantation for Friedreich's ataxia. We weren't the first ones to try this. It's not been tried in patients, but in mice, it works pretty well, surprisingly well. Friedreich's ataxia is not a metabolic disease. It's a mitochondrial disease. And it works. I don't know why. And so we're not talking about cross-correction from an enzyme. We're talking about mitochondrial improvement from a microglia to another. I mean, it's not perfect, right? But it does this. The mice do survive longer. They gain weight better neurologically. They're better. So there are ways in which these cells can help other cells, not just by secreting enzyme, but potentially sharing organelles, so make sure microglia. Transplantation is being tested for cystinosis, which is a membrane protein, so the protein is not being secreted. So somehow pieces of lysosome protein are being shared between these cells. And it's also, the standard for X-linked adrenoleukodystrophy, which is also a membrane protein. So it's not working by secreting. So there's a lot more to this idea of cross-correction that can generalize the potential impact of transplants if we learn to do them safely.
Awesome, thank you. And another question that came up is,
I
are
don't
there
know if that
others?
Maybe I just went off a tangent, but
No, that's perfect. There is a question about if there are other safe harbors other than CCR5.
yeah, you can come up with a list. There are these genes in our genome that you could you do well without, the RHD factor, it depends on I like CCR5 because, I thought it was pretty well established sort of from the human genetic data that it was a safe harbor, but we had several. You can think of like universal safe harbors, genes that people who don't have them are completely fine. You can think of tissue specific safe harbors. Like for a while, my postdoc PI tried to use the hemoglobin locus to express something in the brain. So you maybe a gene that's not leading that particular compartment. So yeah, there's many options for safe harbors.
Awesome. And I think we have time for another one here. And you touch on a couple of times hurdles with FDA and how you make people understand and actually approve things, right? So what is your experience dealing with this paperwork and type of data that's needed for this type of work that you have been doing?
Yeah, so I've only done the easy part, which is had a pre-IND meeting. I haven't had a kind of an IND meeting, which is harder, or the next stage, which is the approval part. But I think it's pretty significant for someone at my stage. I'm an assistant professor, as I've said several times, but I do work with a group at Stanford, the Center of Definitive Curative Medicine, which has provided a lot of help with these other sort of things that go beyond basic, the regulatory part, the clinical. I work very closely with, I actually don't do the transplants. I'm a geneticist to design a clinical protocol. So what I'm saying is just I haven't done this by myself. I needed a team with this other expertise to help me put this together. I think, we submitted a pre-IND package. You have some very specific questions, what you feel like, oh, I don't know what to do. I want to do this. Are you okay with this? Basically, you get like sort of yes or no answers. And, so far, I think the FDA, they were very helpful. I think the comments were reasonable. The only place where we have disagreements and we think that we're going to bridge that is the clinical trial. The FDA feels that we said, well, we'll give this therapy. Let's say the therapy exists. It will be available not for someone who has a perfect donor, let's say an identical, a sibling transplant, for example, right? So those have a perfect standard of care treatment. This is not going to be, because it is an experimental therapy after all. But everyone else could get it. And they said, no, not everyone else. They have to be some like other thing. And then we have to wait until 1 year after they look for other potential donors. And like I said, 1 year seems like a really long time for patients with severe hurdles to be transplanted. So, but we think that, over time, maybe that'll change, and we'll see where that goes.
Awesome. Thank you very much. I think we don't have any other questions coming from the chat here. So I would like to thank you again for joining us. It was wonderful